Dear student, I am Dr. Prakash Mungli, a professor of biochemistry. So in this lecture on catabolism of carbon skeletons of amino acids, especially I will be going over amino acids that form acetyl-CoA or acetoacetyl-CoA. You know, acetyl-CoA or acetoacetyl-CoA as a metabolic fate, they can go into ketone body formation. So basically here I will be going over amino acids that form ketones. So then we can name them as ketogenic amino acids. Let's look into the details. So there are 6 ketogenic amino acids which can either form acetyl-CoA or acetoacetyl-CoA, and in that there are 2 branched chain amino acids, isoleucine and leucine, which can make ketone bodies, and there are 3 aromatic amino acids, phenylalanine, tyrosine and tryptophan, and 1 basic amino acid, that is lysine. So basically we have 6 ketogenic amino acids. So just to write them together here, we have leucine, which is a purely ketogenic amino acid, lysine, which is a purely ketogenic amino acid, and then we have 3 aromatic amino acids, that is phenylalanine, tyrosine, tryptophan. 3 aromatic amino acids are both glucogenic and ketogenic. So here we will talk about ketogenic. And then isoleucine, a branched chain amino acid, which is both glucogenic and ketogenic. And to this list, in my previous lecture, in the amino acids that form pyruvate, in that lecture, I have already told you the confusion that is there in books from different authors about 309, metabolic fate of 309, carbon skeleton of 309. So the carbon skeleton of 309 can either make pyruvate or acetaldehyde, which is making acetyl-CoA, and acetyl-CoA can go into ketone body also. So I'll just add that 309 here also. So in that sense, we have 2 pure ketogenic amino acids, purely ketogenic, that is leucine and lysine, and I given you the mnemonic as Leucine-Lyse, just to remember that, Leucine-Lyse. That's just a mnemonic to remember leucine and lysine, ketogenic. And then we have phenylalanine, tyrosine, tryptophan, isoleucine and 309. These are all both glucogenic and ketogenic. Since we are looking at ketogenic aspect, so we are adding all these here together. So these are the amino acids that will make ketone bodies via going through acetyl-CoA or acetoacetyl-CoA. That is what is shown briefly in this particular figure, where isoleucine via 6 steps will make propionyl-CoA, and the propionyl-CoA going into succinyl-CoA, and succinyl-CoA is it can go into glucose formation. That's a glucogenic side of isoleucine. And also isoleucine make acetyl-CoA going like this, right? So that's how it is ketogenic. Similarly, leucine, as you see, leucine is only ketogenic. Phenylalanine is both ketogenic and glucogenic because it makes fumarate there. You'll have to go one by one with each amino acid. Okay, be careful there. Phenylalanine going into tyrosine, so that covers 2 amino acids, phenylalanine and tyrosine there, which are both glucogenic and ketogenic. In the coming to tryptophan, tryptophan will make quinolinate. Quinolinate makes nicotinic acid. That's the special product coming from tryptophan, catabolism. And tryptophan can make alanine. Alanine is glucogenic, remember. And then on the other side, it makes alpha-ketoadipate, and that alpha-ketoadipate will make acetoacetyl-CoA, which will become acetoacetate, which is a ketone body. Lysine makes alpha-ketoadipate and making acetoacetyl-CoA, and acetoacetyl-CoA making acetoacetate. That's a ketone body. So like that, and the threonine will make acetaldehyde and acetyl-CoA, which will go into keto side, and threonine making glycine to serine, and serine going into pyruvate. That's the glucogenic side of threonine. So like this, we have leucine and lysine, phenylalanine, tyrosine, tryptophan, and then isoleucine and threonine. These are all ketogenic amino acids here. Now let's move on to see how isoleucine is ketogenic here. So the pathway that is shown in this particular slide, just pay attention to how isoleucine is going into ketogenic side. So pathway that starts as leucine, isoleucine and valine. These are 3 branched chain amino acids, as you all know. So all of them undergoes transamination reaction. So because the initial steps are strikingly very similar for all 3 branched chain amino acids, leucine, isoleucine, valine, all of them going through transamination reaction, becoming their corresponding alpha keto acids after transamination, and their corresponding alpha keto acids will undergo oxidative decarboxylation reaction catalyzed by branched chain alpha keto acid decarboxylase enzyme, and then they become their corresponding acyl-CoA thioesters, and these acyl-CoA thioesters will undergo dehydrogenation process, making corresponding alpha beta unsaturated acyl-CoA thioesters, and then the diversion goes. That means from isoleucine there will be formation of beta hydroxy, beta methyl glutaryl-CoA, which will make acetoacetate and acetyl-CoA, and that means leucine is only ketogenic because it makes acetoacetate and acetyl-CoA. Whereas isoleucine, as you see, it makes propionyl-CoA and acetyl-CoA, and propionyl-CoA is glucogenic. Propionyl-CoA making succinyl-CoA, it can go into glucose formation, whereas acetyl-CoA will make acetoacetate, and acetoacetate is a ketone body, it's ketone. That's how isoleucine is both glucogenic and ketogenic. So we are looking at the amino acid that makes ketones. So isoleucine makes ketone. On the other side, we have seen leucine is purely ketogenic because it makes acetoacetate, which is a ketone body itself. Acetyl-CoA can go into acetoacetate formation. So that's the ketogenic. Valine, look at valine. Valine makes succinyl-CoA, and succinyl-CoA converted to propionyl-CoA, and propionyl-CoA can make glucose. That's why valine is only glucogenic. So like this, so our intention here was to see how isoleucine is going all the way to ketone body formation, that is through acetyl-CoA, and acetyl-CoA going into acetoacetate. So that's how isoleucine is ketogenic, of course, and also it is a glucogenic. So isoleucine is both glucogenic and ketogenic. So we are only looking at the ketogenic part of that. Alright. Now here I have written the brief summary of what I have explained in the previous slide. So isoleucine undergoes initial transamination reaction followed by oxidative decarboxylation reaction, and downstream oxidative reaction yields, importantly, succinyl-CoA and acetyl-CoA. And this succinyl-CoA, it will make propionyl-CoA, and that makes glucose. That's the glucogenic there. Acetyl-CoA will go into acetoacetate formation, and that is ketone body. That's why isoleucine is both glucogenic and ketogenic amino acids. Remember that point. Now let's move on to see a metabolism of leucine, which will make a ketone body, which I have already explained you in the previous slide. So leucine undergoing transamination reaction, oxidative decarboxylation reaction, and downstream oxidative pathways will give a hydroxymethyl glutaryl-CoA, which will make acetoacetate, which is a ketone body itself, and acetyl-CoA in turn, it will become acetoacetate, that is a ketone body. So that means leucine is a pure ketogenic. Leucine is purely ketogenic amino acid. Remember that. So that's what I have briefly written in this slide. Leucine undergoes initial transamination reaction followed by oxidative decarboxylation reaction and then downstream oxidation reaction to yield acetoacetate and acetyl-CoA. And acetoacetate is a ketone body itself, and acetyl-CoA may go into ketone body formation, that is acetoacetate. So leucine is only ketogenic amino acid. It is only ketogenic amino acid. That's how our leucine goes into ketone body formation. So now let's move on to see how phenylalanine, tyrosine will go into ketone body formation. So the figure here, which is showing the phenylalanine converted into tyrosine by enzyme phenylalanine hydroxylase, and the phenylalanine converting into tyrosine, further tyrosine goes into p-hydroxyphenyl pyruvate, and the p-hydroxyphenyl pyruvate formation, it will be done by tyrosine aminotransferase enzyme, and then p-hydroxyphenyl pyruvate will be converted into homogentisate, and that will be done by p-hydroxyphenyl pyruvate dioxygenase, which is also sometimes referred as 4-hydroxyphenyl pyruvate dioxygenase, and the homogentisate further converted into malonyl acetoacetate, that will be done by homogentisate 1,2-dioxygenase, which is also called as homogentisate oxidase. And then the malonyl acetoacetate will be converted into fumaryl acetoacetate, and then fumaryl acetoacetate will be converted into fumarate and acetoacetate, done by fumaryl acetoacetate hydrolase enzyme. And note that this malonyl acetoacetate and fumaryl acetoacetate, they can go towards succinyl acetoacetate formation, which further forms succinyl acetone. So like this, phenylalanine and tyrosine will make both fumarate and acetoacetate ultimately. Okay, let me just highlight that one more time so that you are aware what all the important points that you must be knowing in the catabolism of phenylalanine and tyrosine, especially paying attention to the fate of carbon skeleton. It is phenylalanine converted to tyrosine. That particular reaction is catalyzed by phenylalanine hydroxylase enzyme. Note that this phenylalanine hydroxylase, it needs a coenzyme called BH4 tetrahydrobiopterin, and the deficiency of tetrahydrobiopterin or defect or absence of phenylalanine hydroxylase will give rise to different types of phenylketonurias like type 1, type 2, type 3 and type 4. We are going to go into the details of phenylketonuria. So that's one of the clinical application that we should aware with this particular reaction. Conversion of phenylalanine into tyrosine done by phenylalanine hydroxylase. After that, tyrosine is undergoing a transamination reaction done by tyrosine aminotransferase to become p-hydroxyphenyl pyruvate. Deficiency of tyrosine aminotransferase may lead to a transient tyrosinemia, and then p-hydroxyphenyl pyruvate undergoing a dioxygenase catalyzed reaction, that is p-hydroxyphenyl pyruvate dioxygenase to become homogentisate, and the deficiency of 4-hydroxyphenyl pyruvate dioxygenase will also lead to neonatal tyrosinemia. Now the other important reaction that you must be aware here, the most high yield one apart from phenylalanine hydroxylase one, of course phenylketonuria is important. So homogentisate is converted to malonyl acetoacetate done by homogentisate oxidase, which is written here as homogentisate dioxygenase. can be written as homogentisate oxidase, very important enzyme. Defect or deficiency in this enzyme will give rise to alkaptonuria, very interesting condition with so many interesting features there, characteristic findings for the diagnosis. Now, in this disease, because of deficiency of homogentisate oxidase, there will be accumulation of homogentisate, and this homogentisate will be getting into alternate metabolic fate to become benzoquinone acetate. And this benzoquinone acetate will have a higher affinity for cartilaginous tissue, and it's going to stain the tissue. We are going to look into that aspect in alkaptonuria disease. Now, moving on, malonyl acetate undergoes isomerase catalyzed reaction to make fumaryl acetoacetate, and the fumaryl acetoacetate undergoes a reaction catalyzed by fumaryl acetoacetate hydrolase enzyme to break into fumarate, and fumarate is glucogenic. That's why it is a phenylalanine and tyrosine are glucogenic amino acid, and also acetoacetate itself is a ketone body. So that is why phenylalanine and tyrosine are both glucogenic and ketogenic. So this is how phenylalanine and tyrosine makes acetoacetate. That's where we are in here, because we are looking at catabolism of carbon skeleton of amino acids that make either acetyl-CoA or acetoacetyl-CoA, making acetoacetate as the ketone body. So we have seen that now. Just to give the clinical application of the last reaction that we have seen, conversion of fumaryl acetoacetate into fumarate plus acetoacetate done by fumaryl acetoacetate hydrolase enzyme. Deficiency of this enzyme, it will give rise to a disease called type 1 tyrosinemia. And type 1 tyrosinemia is also referred as tyrosinosis. Okay, in this disease, there will be accumulation of fumaryl acetoacetate because fumaryl acetoacetate hydrolase is deficient and the reaction is not going on sufficiently. And when there is a buildup of fumaryl acetoacetate, so it can also go towards formation of malonyl acetoacetate. Both of these molecules will accumulate and they will undergo alternate metabolic fate to become succinyl acetone, succinyl acetoacetate, and becoming succinyl acetone. And this succinyl acetone, it can lead to kidney damage and liver damage, because succinyl acetone is a mitochondrial toxin. It will have a toxic effects on mitochondria, and that's why it lead to liver damage and kidney damage. Okay, so these are some of the clinical things that we are going to go over later. So at this point now, I wanted to bring to your attention that phenylalanine and tyrosine upon catabolism will make acetoacetate and fumarate. Acetoacetate is ketone body itself, fumarate is, it can go into glucose formation. So phenylalanine and tyrosine are both glucogenic and ketogenic. So while I was explaining that, I was kind of introducing you to the clinical aspects of this metabolic pathway. Some of the clinical disorders that we are going to go over later is phenylketonuria, and then we are going to go over alkaptonuria, then we are going to go over type 1 tyrosinemia, and briefly we will go over transient tyrosinemia, neonatal tyrosinemia. These are some of the disorders that we are going to go over later. So in this slide, I have briefly given you the explanation for conversion of phenylalanine and tyrosine into fumarate and acetoacetate. So phenylalanine and tyrosine undergoes initial hydroxylation and transamination followed by downstream oxidative reactions to give fumarate and acetoacetate. Fumarate via malate become glucose. Acetoacetate is a ketone body. So both phenylalanine and tyrosine are ketogenic and glucogenic. Now let's look at the last amino acid which makes acetyl-CoA or acetyl-CoA, making ketone body, that is the tryptophan. Tryptophan undergoes 8 reactions to release alanine and alpha keto adipate, and the alpha keto adipate undergoes 6 reactions to give 2 acetyl-CoA. These 2 acetyl-CoAs will give acetoacetate, and acetoacetate is a ketone body, and that's how, and alanine can go into glucose formation, and that is how tryptophan, by making alanine it can make glucose, and by making acetyl-CoA it can make acetoacetate, which is a ketone body. Okay, so this is a very brief outline of the metabolic pathways. There are multiple reactions involved, but is the most simplified way of explaining you how tryptophan is going into glucose formation and also ketone body formation. So remember the intermediates. Tryptophan will give alanine and alpha keto adipate, and alanine goes into glucose formation in gluconeogenesis process, which you already know. I have already explained you in my earlier lectures about gluconeogenesis process, how alanine exactly converted to glucose. And alpha keto adipate undergoes 6 more reactions, oxidative reactions, to make acetyl-CoAs, and that these acetyl-CoAs can go towards ketone body formation. Okay. So here in this slide, I have briefly explained you now how tryptophan is both glucogenic and ketogenic. So tryptophan upon oxidation will yield acetyl-CoA and alanine. Acetyl-CoA may form acetoacetate, which is a ketone body. Alanine undergoes transamination reaction to form pyruvate, and pyruvate will go towards glucose formation. So tryptophan is both glucogenic and ketogenic. And finally, the oxidation of lysine is very similar to tryptophan to yield acetyl-CoA, which will go into ketone body formation. So lysine is only ketogenic. So that covers the fate of lysine to go into ketone body formation. So with all this, I have explained you how all these amino acids will make in one or the other way ketone bodies. So just to name all of them now together. So we have leucine, lysine, phenylalanine, tyrosine, tryptophan, isoleucine and threonine. All of them can make ketone bodies. So within them, leucine and lysine are purely ketogenic amino acids, and then 3 aromatic amino acids, that is phenylalanine, tyrosine and tryptophan, and then along with that a branched chain amino acid isoleucine and a hydroxyl containing amino acid threonine. They also make both glucose and ketone body. Okay, so there are 5 amino acids which are making both glucose and ketone body, glucogenic and ketogenic, and 2 amino acids will make only ketone bodies. Those are leucine and lysine. So this is all about catabolism of carbon skeleton of amino acids, which will go into ketone body formation. Thank you.